Good morning, everyone. I am Huma, and I'm a PhD candidate. Today, I'm going to talk about one part of my PhD thesis, which is about analysis of microRNA produced by PPC-tolerant species reveals the novel target gene in Fusarium circinatum. Talking about a little introduction, Fusarium circinatum is one of the world's most invasive and virulent pathogen, which is causing infecting almost 106 plant species, which is including 67 species of pines. This disease has come in Europe from the USA, and it's still present in Aberrania persinola, including Spain and Portugal, which has been eradicated from other parts of the USA and other continents. This pathogen is famous for causing a disease which is known as pine pitch canker because it's causing the cankers in the different part of the plant. It's not only producing the problems in the nurseries, but it has drastic effects on the trees' mortalities in the forest range. So up till now, from 2003, when it has been came here in Europe, we can't control it effectively. So still, we didn't find any effective treatment for this disease. I would like to call you something that we studied in our secondary education about the central dogma, in which we have studied, like the DNA, they can transcribe into the messenger RNA, and this messenger RNA is translated into the protein. The protein, no proteins mean non-translation. So, talking about something about the RNA interference, this is basically a natural mechanism as from the past few decades it has been studied, like we don't need to use the fungicides to control the forest pathogens, so we have to use an innovative potential approach or a biological process to control the disease. So this is basically, we use a form of RNA interference, which is also known as gene silencing mechanism. If we're going to tell you about what is RNA interference, so just imagine we have a double-stranded RNA in our cells, and then it is going to cleave into the small RNAs. These small RNAs can be in two forms. One is microRNAs that are produced naturally in the cells, and small interfering RNAs. These small interfering RNAs are different because they are produced when we are going to apply DSR RNAs against pathogenic genes. So what they do, these small RNAs go inside the cell, and with the help of RNA machinery, which is conserved and present in all eukaryotic organisms, it goes and bind to the messenger RNA with the complementary sequence, and what they did, it chop off the messenger RNAs, and this chopping of the messenger RNA resulting in no protein. So it means this silencing mechanism can be used to control the pathogenic genes. I'm going to tell you a very interesting fact that it has been found, this communication, like the RNA interference molecules, which are small interfering RNAs or microRNAs, they can be communicate with each other, which is also known as cross-kingdom communication. It means like these small RNAs can be transferred from the pathogen to the host and from host to the pathogen. Well, in our lab, we are still working on that, so I'm just going to give you the examples from the literature. Like in 2019, Kai and their colleagues, they have studied that the host, which is any plant that can be any host, but they have studied with the Arabidopsis, which is a model plant, that these Arabidopsis, they send small they receive small RNAs from a host, which is a Botrytis cinerea, and this pathogen has infecting more than 1,000 species of plants. And these small RNAs goes to the host, and it hijacks the immunity of the host. And they found that these small RNAs in the host, they affect all the genes that are related to the signaling molecules of the host, and also all the genes that are present in the cell walls. And we can see the symptom development, the one in the host that we're targeting and the one with the wild type. Well, this is not finished here. They've also found that, okay, host can receive these small RNAs from the pathogen, but host can also send the small RNAs to the pathogen to inhibit its virulence. So they have studied the same pathogen and the same host, and they have discovered that when we are going to knock down the genes of the pathogen, for example, these genes are related to the vesicle trafficking, and we have found that these vesicle trafficking genes inhibit the virulence, and the symptoms are less as compared to the wild type. Taking into account this literature, we have tried to figure it out in our lab with the pathogen, which is Fusarium circinatum, to use it to look for the new target genes, and we use the data from the microRNAs. But before that, I'm going to tell you what are microRNAs and how they are going to produce. MicroRNAs basically are naturally occurring molecules that are non-coding, and they have length of 18 to 28. And the good thing is about that, there's only present in eukaryotes, so we can use them to target the pathogen. They can serve as a gene regulator, and one microRNA can control more than one messenger RNA. It means we can use the one microRNA against more than one genes at a time. They can target messenger RNA by the complementary sequence, as I told you before. They can involve the messenger RNA degradation, not in the animals, but also in the plants. So we can use more than that. And they have, as I told you, like they have involved in the cross-kingdom communication, like the microRNAs can transfer to the plants, and the plants can also send microRNAs to the pathogen to inhibit the virulence. Well, in the case of biogenesis of microRNAs naturally, we have found that they can't produce like in the form of mature. The microRNAs, first of all, primarily described into the primary microRNAs, which have a loop structure like this, and this loop structure with the help of RNase enzymes, they are broken down into the precursor microRNA. These precursor microRNA basically are not mature, so they can't bind to the messenger RNA. So they need to be broken down with the help of RS, which is RNA machinery enzyme that I told you before, that is conserved in eukaryotics, and they form a mature microRNA, and these microRNA are going to target the messenger RNA and then chop off the protein. Well, it's not studied for the first time, like it was discovered like in 1993 in nematode, it has been found. And the interesting thing is that some of the microRNAs are conserved between the different species. For example, some microRNAs also present in the fly, and the same microRNA we can also find in the humans and also in the nematode. So this is one of the interesting facts, that if we knew some microRNAs that are conserved, we can also use these microRNAs against different pathogens to control. Well, we did an experimental approach in our lab to study these microRNAs to look for the target genes, and we used two different types of different species. The one species, which is Pinus radiata, which is the susceptible one, and the one is tolerant one. Why we use the both species? Because we want to know either the tolerant can produce more microRNAs as compared to susceptible, because tolerant is always resistant to a pathogen, so it should have any defensive approach against this. We do the extraction of microRNAs, and then we send these for the sequencing. And what we found? We have found the different count and expression of these microRNAs in both different species of the pine, like in also the tolerant and also in the susceptible one. And we have found two different microRNAs. One is the non-ones and one is the novel ones. The novel means these are already described in the reference genome, and this reference genome was Pinus radiata. Next, we did this validation of these microRNAs in our lab with the help of qRT-PCRs, because the expression are very low, and sometimes we have to further confirm the expression and count from the sequencing. And what have we found? That out of the 49 that are total, we have 37 non, and the 12 are novel that are just found in our host against our pathogen. Then we do another validation to find out either we have any difference between the tolerant and in the susceptible, and we get the good results, like 30 of the microRNAs are tolerant in our pathogen and 10 are susceptible, and well, 9 microRNAs are common in both the species. And the good fact is that, I have told you before, the length should be around 18 to 28 of the microRNAs. Then we can see, okay, these microRNAs are the good ones. So we have identified most of these microRNAs are 21, and then they are followed by the 20, 21, 22, 19, 23, and 18 nucleotide. That we are safe, and these microRNAs can be used for looking for the new target genes. Next, we do the expression and how we look the new target genes. We found out the data, and we have found that one of the non and the two of the novel microRNAs, they are present in our tolerant species, but not in our susceptible species. So what we did, because we know we have a lot of data, and we want to know either the microRNA is going to affect our genes of the Fusarium cercinatum, so we go to an online plant database where all the plants data are present, where that first produce the microRNAs against any pathogen. And we did the blast, and we have found like 28 genes per microRNAs against our Fusarium cercinatum transcriptome. And all these genes are very important because they are related to signaling, virulence, metabolic process, different cellular respiration process. And then we select few of these genes so that we can validate their virulence in our pathogen. These genes that we are selected are basically on the basis of the transcription factors, because transcription factor is one of the most important factors to regulate the transcription in the proteins and RNAs. And we also choose some genes that are according to the signaling pathways. Still now, I'm working with this, and I'm now doing the knockouts, and I need your prayers to do that because it's a very difficult process. Well, my next steps will include knockout of genes, so I can see either they can do any phenotypic and pathogenetic change in the pathogen, and I will design new molecules, so I can replace the essential pathways, because the essential pathways are usually conserved in the pathogen, and so I want to use these novel target genes to go for the further silencing process, for example, spray-driven gene silencing or the virus-induced gene silencing. Thank you so much.